data. 2% of the human genome is coding DNA, and 98% is non-coding DNA. Okay, so this non-coding DNA is just the fragments between genes, but you should remember that the genes are split into exonic sequences, which are coding sequences, and intronic sequences, which are not coding sequences. So in this 98% is also this intronic sequence included. Okay, so people used to call this additional DNA, non-coding DNA, junk, but I will tell you that it's not a junk, and it's a kind of funny cartoons that these fragments are shouting, just because you don't understand, you cannot call us junk, because you will see that these fragments, actually these non-coding fragments can code non-coding RNAs, actually. They can contain genes for non-coding RNAs. And these non-coding RNAs can be divided into two groups. One is housekeeping non-coding RNAs and regulatory non-coding RNAs. This housekeeping non-coding RNAs group includes rRNA, which is ribosomal RNA, important for what? Okay. tRNA, which is transfer RNA, which is important for translation, for bringing amino acids. snRNA, small nuclear RNA, involved in splicing, removing of introns from pre-mRNA, the mRNA precursors. And snRNA is small nucleolar RNAs involved in processing of rRNA. So these two, snRNA and snRNAs, are involved in processing of other RNAs. So in the group of regulatory non-coding RNAs, you can divide them artificially, actually, into short non-coding RNAs and long non-coding RNAs. And actually, as you can see here, you have the basis of this distribution between these two subgroups is the size. So smaller than 200 nucleotides, it means short non-coding RNAs, and bigger than 200 long non-coding RNAs. And in the group of short non-coding RNAs are microRNAs, siRNAs, piRNAs, scarRNAs, and you heard already a lot about microRNAs, and you will hear even in future. Zofia will present some lectures and presentations showing the function of this microRNA. But today, I will concentrate only on this long non-coding RNAs, which this group contains linear non-coding RNAs, circular RNAs. eRNA means RNA transcribed from enhancers. Do you know what enhancers is? Enhancer is a special regulatory sequence which enhances transcription. Okay, we will see, we will discuss the mechanism of it today. And NAT, it means natural antisense RNA. So it can be that some non-coding RNAs are transcribed from the complementary strand in the same fragment which protein coding genes is encoded. You will see the examples I will explain in details. But let's start from definition. So long non-coding RNAs, definition. So first, we should understand that genomes are extensively transcribed and give rise to thousands of long non-coding RNAs, which are defined as RNAs longer than 200 nucleotides that are not translated into functional proteins. And here we should be really careful, because that are not translated into functional proteins means proteins, but they can sometimes encode smaller peptides. We should remember about it. Okay, so, and this genome, what I said about that genomes are extensively transcribed, it means that from this 98% of non-coding RNA, non-coding DNA, probably 90% is transcribed. So you should have this picture of the genome, that it's not the protein coding gene, protein coding gene, protein coding gene that are transcribed, but that even the fragments between them or some other fragments of, I mean, all genome is, almost all genomes are transcribed. That's really something new view of what's going on in the nucleus. Okay, so this broad definition encompasses a large and highly heterogeneous collection of transcripts that differ in their biogenesis and genomic origin. So we should remember that they are not the same. So different long non-coding RNAs are, I mean, their origin is different, and you will see, and the function and mechanism, I will present today a lot of different mechanisms, and you will see how different these mechanisms are. And statistics from human gene code suggest that the human genome contains more than 16,000 long non-coding RNA genes. Some other people said that it's even up to 100. I think it's a little bit too much. But only really a small group of them are characterized. This is what we should remember, that it's still uncovered field, completely uncovered, not completely, but almost completely uncovered field. Good for you to study, to do your PhD on this, in this subject. In plants, 600 different long non-coding RNAs were identified. It doesn't mean that plants contains less non-coding, long non-coding RNAs, but actually they are less studied. Actually, this is the problem. You know that it's always that human genome is studied a lot and in many laboratories, and that the plants are just in isolated laboratories and groups. So we for sure we have much more than this. Characteristic features now. First, long non-coding RNAs mainly include RNA molecules transcribed by RNA polymerase II, Pol2. Do you know that in eukaryotes you have 3 polymerases? Polymerase II, 1 and 3. 2 transcribes protein coding genes, and as you can see here, most of long non-coding RNA genes. 3 is for 5S RNA and tRNA, and 1 is for ribosomal RNA. So you have a special function for each polymerase. So mainly, I mean, I said that most of RNA molecules are synthesized by RNA polymerase II, but not all, because other polymerases can be involved as well. And RNA molecules from intergenic regions, as well as sense of antisense transcripts that overlap with other genes can be produced. It means that it can contain, they always contain their own genes, but these genes can be between the protein coding genes in the middle, or it can be, they can overlap in different directions. You will see the example, I will show you this. So because in most cases they are transcribed by RNA polymerase II, they are kept by 7-methyl guanosine M6G at their 5' ends and polyadenylated at their 3' ends. You know what the cap is? A special structure at the 5' end of mRNA, and poly A is a special structure at the 3' end, important for what? Do you remember? Boom. Translation, and yeah, exactly, and stability also, and export. You should remember that in all this stability, export from the nucleus to the cytoplasm, and translation. What's really interesting, they can contain introns that are spliced similar to mRNAs. So they're kind of similar to mRNAs. What's really important is what I said before, that enhancers and promoters are also transcribed. This is something new, and we discovered this during global sequencing, this high-throughput sequencing, because during sequencing we discovered that all genome is transcribed, almost all genome is transcribed. So it was, of course, I mean, maybe I should add this, that transcription of protein genes is at the very high level, or sometimes they are also silenced, but if the transcription is on, it's a high level. But transcription of enhancers or transcription of promoters, sequences from promoters, are at the lower level. So before high-throughput sequencing, we couldn't detect these transcripts from enhancers or promoters, but because of the new techniques, we can do it. And you have even the special names for enhancer RNAs, eRNAs, or you can have promoter upstream transcripts, prompts. This is a kind of, no one knows actually what it is. It's just polymerase is transcribed the gene from one direction, but then sometimes they are the same polymerases transcribes the second strand from the promoter. This is very short fragments, and all these fragments are degraded very fast. Okay. What, in general, before we will discuss each case and which long non-coding RNAs and its involvement in transcription, gene expression regulation, some general feature. Long non-coding RNAs contain some repeat domains, and these domains are important for. First, some sequence is complementary to DNA at certain position and can form what we call R-loop. R-loop is a special 3 strands structure where RNA, here, for example, it's Apollo, long non-coding RNA, hybridize with one strand of DNA, and the second strand is displayed. So we have this bulge with RNA complementary to DNA and another strand of DNA, and this is called R-loop, very important for action and function of non-coding RNAs. Long non-coding RNA can contain some motifs or sequences. They are recognized by some proteins involved in other processes, and these proteins can bind to this long non-coding RNAs, and of course cannot work in this situation. It means that the long non-coding RNA can sequester the proteins and stop somehow action of it. Of course, this is another possibility, is that it's not, the protein will not recognize the sequence like here, YYU, CU, Y, but just the structure of the RNA. Or it can be involved in RNA-RNA binding. You will see the example with Stauffen-1 protein, which you will see that the long non-coding RNA, which can base pair with mRNA. And this is a stimulated by Stauffen, you will see this action. So in all these cases, the most important part is binding of 2 partners, protein partner, DNA, of course, the sequence, or the RNA partner in the last example. And then I have to now I have to introduce the circular RNA, because circular RNAs are actually products of so-called back splicing. So you can see splicing of regular pre-mRNA exon. You have exon 1, 2, 3, 4 introns, and these introns in regular splicing are removed. You know probably this from some lectures. And it's catalyzed by the spliceosome. The spliceosome is a huge machinery built of hundreds proteins and snRNAs, actually, small nuclear RNAs, which we mentioned. And this spliceosome is important for for the splicing. It means removing introns and putting exons together into mature mRNAs. The same is true in case of some long non-coding RNAs. So as I said, some non-coding RNAs contain introns, and this intron must be removed in the same way. Okay, but the same spliceosome in some cases do really strange thing, which is called back splicing. It means instead of connected this splice site with this splice site and remove the intron, they do the back, somehow the opposite reaction, and joins this site with this site, forming a circular RNAs. I'm sorry. And this circular RNAs can contain exonic sequences, like here, because this intron can be even removed from the circular RNA. Or they can have one exon, two exons. Sometimes even they contain introns. So they can be really heterogeneous. And they actually will work exactly the same way like long non-coding RNA. And just to show you this circular RNA, you can see here that in this picture you have exon intron, the circular RNA with exon intron only, for example, only exonic, two exon RNAs, and then function. First of all, it can bind proteins. So this is exactly what I said before. It can bind RNA, and the most cases, this circular RNA will bind microRNA. We call it that it's a sponge for microRNA. A sponge because it takes microRNA from its their action in mRNA's digestion or translation inhibition. It means that if you express in the cell this circular non-coding RNAs, you will not observe the action of certain microRNAs because the sponge will absorb, or not absorb, make a complementary sequence. I mean, by complementarity, will do this base pairing and then removed microRNAs. The same the long non-coding RNAs can do the same. And this is very important function of non-coding, in general, long non-coding RNAs. So protein, RNA, in this case, in most cases, 90% probably it will be microRNA, and surprise, surprise, protein translation. This is a really weird situation that because we have a coding sequence here, in many cases, ribosomes can start translation of this. The mechanism is unusual and very strange because to start translation, you need the cap structure in eukaryotes because the ribosome will recognize the cap structure first and then will scan to find the AUG codon and then start translation. But here, you don't have a cap structure because it's circular. So you must have another mechanism. This is a big debate and discussion, scientific discussion, how it happens. We know that this is for sure, they can be translated. So now it's completely mess. Non-coding RNAs, circular, translated into proteins and blah, blah, blah. So they are coding, non-coding RNA, coding protein, non-coding RNAs. So this field is really new and very dynamic, and this is why you have this kind of, a lot of this kind of situations. Okay. Some information about biogenesis and cellular fates of long non-coding RNAs. Transcription is exactly the same like mRNAs, like protein coding genes. So usually you have a promoter, exons, introns. Of course, here you will not call, you can call them exons, but because they will put together, an intron will be removed. But this, in case of long non-coding RNAs, these exons are not coding protein. So what's characteristic for long non-coding RNA transcript is that they are usually degraded in the nucleus. So the splicing of these introns in long non-coding RNA precursors is very slow and inefficient. So it means that they usually, a lot of them will be degraded in the nucleus, and only a small proportion will be exported to the cytoplasm and in the cytoplasm. Degradation, the mechanism of nuclear retention, so it means that why these products, these substrates, and these transcripts are retained in the nucleus is like this. So very often, even during the synthesis of this non-coding RNAs, exosome, I mean this is a degradation complex, will degrade them. So it's a system controlling that if something like this will appear, will be degraded. But in some cases, they will be kept close to the transcription sites on chromatin by binding with some proteins. And this is important because you will see that this is very crucial for action of long non-coding RNAs. What's really interesting is that you want snRNA, one of this snRNA which is involved in splicing, is involved in tethering of non-coding RNAs on chromatin. So you have a binding of, some special binding of this snRNA, actually with proteins, an snRNA with proteins called snRNP, RNA protein complex. And this is why it's not degraded, you do not see degradation of this long non-coding RNAs, but you see tethering on chromatin. Then you have weak splicing signals, what I said, and these splicing signals can stop actually splicing and export of this long non-coding RNAs into the cytoplasm, and some other signals that can, let's say, some special proteins complexes can bind here and then support, not support, but block splicing of this intron. So you have a lot of different methods, or not methods, but approaches to keep this long non-coding RNAs in the nucleus. So you can see immediately that the main action and the main function of this long non-coding RNAs will be in the nucleus. But sometimes they are exported and they can bind some proteins in the cytoplasm. They can influence translation even. You will see this some example. And surprise, surprise, can go to the mitochondria to work in mitochondria inside, or they can be exported from cell to cell using exosome system. Exosome is a kind of vehicles, bubbles to transfer, to get something from one cell and then transfer to another cell. Okay? So this is really new stuff and very interesting, actually. Mitochondria, you can see that I put here question marks because it's not clear actually how it works and why it's like this and so on. Okay, so now we know how long non-coding RNAs are produced. And then we can focus on functions. And we are interested in the regulation of gene expression, and they can do this through they participate in general nuclear organization. They will model chromatin structure. They can influence directly transcription or control post-transcriptional RNA processing. And we will see one after another all this possibility in several examples. Let's concentrate on this participation in general nuclear organization. What I mean by this. This is the nucleus, and you know that in the nucleus you have a chromatin, of course, of different chromosomes, but you have a lot of different structures. For example, you know the nucleolus, for example. This is the place of, what's the role of nucleolus in the cell? Of RNA, so it's a production of ribosomes, because it's not only synthesis, it's processing of this, and then ribosomal proteins are going from the cytoplasm to the nucleolus. They bind to rRNA, forming subunits of ribosomes, and subunits of ribosome are exported to the cytoplasm. So it's a very important center of rRNA transcription plus assembly of subunits of ribosomes. Okay, but we'll not talk about this, guys, but you have plenty of different structures, foci, thank you, foci. And this foci contain usually long non-coding RNAs and a lot of proteins. Here you have examples, the best known examples are nuclear speckles. In nuclear speckles, a lot of splicing factors are accumulated. And this is possible only because long non-coding RNA mallat is expressed, and it stimulates formation of this kind of structures. You have paraspeckles. It's a different structures, bodies attached to speckles, as you can see here. You can have perinucleolar compartments, speckles, and all other structures. We will not discuss in detail the composition of them, but I would like to stress that in all cases you have long non-coding RNAs which will stimulate formation of this. How? Just the long non-coding RNAs with binding sites for proteins. Proteins will bind to it and will condensate, and then will produce this kind of structure. These structures are membraneless, and it's formed based on this very famous or fancy, sexy right now, liquid phase separation. So this is the protein some proteins where when they are very close together, they form this kind of jelly structure. I don't know how to describe it, but it's just the condensation of that. But RNA is all cases important for forming this kind of structures. And that's very important part. I mark even in red here. This is in the case of fire, long non-coding RNAs. It's important to get interaction between different chromosomes. So it means that this long non-coding RNAs is required to organize actually chromatin structure inside the nucleus. This is really important because chromatin cannot be mixed together, chromosome 1, chromosome 5, kind of one big mixture. You have to organize everything to make transcription and then later replication easier. So this RNA is important to contact some chromosomes, some chromatin, and then takes place in organization of this chromosome inside the nucleus. So this is why I said that these long non-coding RNAs have roles in general nuclear organization, which is important for normal transcription. This is why I put this example, because organization of the nucleus, it's crucial actually for transcription. If the organization is bad, you can have 100 microRNAs and transcriptional factors, and they cannot control actually anything. Okay, so now let's discuss modeling chromatic structure. So we will see about we will talk about epigenetic regulation. What's epigenetics? What do you think? Do you know what's genetic and epigenetic? Genetic changes will be mutations. Exactly. So it's just not mutations, but changes introduced by modification of chromatin structure, or DNA actually, because we can have DNA methylation is epigenetic mark, but histone modifications are epigenetic marks as well. So you can have both DNA changes and histone modifications. So let's check several examples, and you will see here. Please think about how different these examples are and how many things we have to do to understand the action of the final action of long non-coding RNAs. This is the example. If without any sign here, it means that the example is from humans. When you see nice Arabidopsis here, it means that this is a plant example. So this is human. And we are talking about hot tip long non-coding RNA. It's transcribed. It's bound by some protein, the name is not important, which can recognize another protein, and this complex form a loop, functional loop. So it means that, functional loop means that chromatin is like this, and then if you have enhancer here, this enhancer will work on all these genes which are in the loop. So the loop is this looping is very important for expression. And then the protein of the complex can change histone 3. It means methylated lysine at the position 4 of histone 3, making these genes in this loop active. And these genes are HOXA genes. You have 4 genes like this, and so it means that by introducing the modification of histone H3, the transcription of HOXA genes are on. Okay? Without this long non-coding RNAs, there is no modification of chromatin at this part of the genome, and there is no transcription. One example. So again, the long non-coding RNA is involved in putting some marks on chromatin. The second is very similar, but kind of opposite. Again, it's a synthesis of Pres1, again, from humans, no Arabidopsis here, and it can bind SIRT6, which is deacetylase. Maybe I should explain that acetylated histone 3 means active gene. So if the acetylation of this position, you said CK56 means lysine 56 and lysine 9 is acetylated, the gene is very actively transcribed. But if the acetylase will bind to it and remove acetyl group, it will be blocked. Okay? So in this case, Pres1 not attract modifying enzymes, but actually, yeah, that's good, but it doesn't work in positive way, but in negative way because it binds the acetylase and keeps this active, the gene active. And this is actually a very interesting example because this example is from stem cells, and all these pluripotency genes in stem cells are under control of this Press1 long non-coding RNA. So you can now realize how important long coding RNAs are. Press1 will control expression of genes in stem cells. And stem cells must, you know, be active to divide and replication and so on. So this is really important. If it's not active, the stem cells will be developed into some other type of cells. So it's crucial, Press1. Okay, so this is 2 examples. And now it's example with more connected with long non-coding RNAs, which will bind directly to DNA, to chromatin. So it means, look at these genes, the name is not so important. If DNA is methylated at the promoter region of this gene, the gene is not transcribed. If it's unmethylated, it's transcribed. So what's going here? We are talking about TARID, long non-coding RNA. By the way, these long non-coding RNAs have very often very funny names, actually. I don't know why it's like this, but they have peanuts, TARID, press, and something like this. So TARID, and you can see that TARID is transcribed from this direction, and if here can form a loop structure, R-loop structure. You remember that the complementarity to DNA open the structure. This heteroduplex DNA RNA is recognized by a set of proteins. We will not discuss the names of them. And this set of proteins can demethylate promoter regions of TCF21 gene, and it's active. So it means that transcription of the long non-coding RNA inside, actually very close to the target place, can change methylation and activate transcription of the target gene. Okay, so this is something in which the direction of transcription is very important. The gene must be close to the target gene, and so on. And finally, now it's Arabidopsis. It's, again, will be with direct interaction with chromatin, but actually the action of this this long non-coding, I'm sorry, I'm saying microRNAs because usually I'm talking about microRNAs. Apollo is a very famous long non-coding RNAs in plants. It's induced by auxin and it will induce expression of some target genes. But what's going on? Without Apollo, this Apollo target genes, doesn't matter what kind of, will not name them, they are inactive because the histones are modified in such a way that they are not active. This is important for this methylation is LHP1, which is polycomb factor. The name is not important. So without long non-coding RNAs, Apollo, this locus is inactive because of all this modification in the promoter sites. Maybe I should, you should be really careful because I'm talking about once methylation stimulates, of histone 3 stimulates transcription and in one cases inhibits. Be careful. It depends what kind of lysine and how much methyl groups are added. So this is not so easy that when you have methylation, so when you don't have methylation, when you have acetylation, it's this. You must be really careful. This so-called histone code is not so easy to understand. Okay, so here, if Apollo appears in the cell, they can bind, look at this, R loops. They can bind the polycomb factor, which was important for silencing of target genes, and the target genes are transcribed. So this is similar situation like the last time. Look at this. It's very similar situation like last time, but this Apollo gene is far away from the target genes. So we can say that this gene regulation is a trans. Do you remember what's trans regulation and cis regulation of expression? Yeah?
Cis regulation refers to basically get something near the origin and in the system.
Exactly.
Trans regulation refers to something like it's out of the system, like in the environment.
Okay, yeah, exactly. So transregulation is just another molecule from another site produced. It can be RNA, can be DNA, not because DNA cannot move like this, but RNA or protein, but encoded somewhere else. And in cis, it's very close to each other. Okay, so in both cases, long non-coding RNAs can work. So then we will describe the influencing of transcription in a direct way. I have many examples here, but I think maybe we'll look at some of them. Look at here. This is target gene inhibition, and this is funny because one long non-coding RNA, I should say, can block expression of one gene, which is, look at this is the direction of long non-coding RNA transcription, and this is the gene. This is the example of overlapping genes I told you. And this is because of some steric problems. If polymerase is going in this direction, this polymerase cannot go. So the transcription of long non-coding RNase ERN will block transcription of this gene. And this is not because of some sophisticated mechanism. This is just, a collision of two polymerases, and they cannot. If the transcription of long non-coding RNase is stopped, then transcription of the target genes is on. Okay, but at the same time, and this is really, so this is a kind of, I would say, cis regulation, but at the same time, the same long non-coding RNAs can act on some other genes by binding protein, which will block chromatin at this position. So see, this is two function of the one long non-coding RNase, ERN, can function on one gene and on the two other genes somewhere else, in cis or in trans. And this is the very important example, very general. This is what I told you, that enhancer can be transcribed, producing long non-coding RNA. And this long non-coding RNA will bind by activating complex and then promote PCG, means protein coding gene, any protein coding genes. And this is actually how enhancers work. So it means that by transcription of eRNA can form activating complex and then can activate transcription. So this is something new. maybe not for you because it was described several years ago, but for us it was a kind of shocking information that the enhancers can be transcribed. Enhancers can be transcribed and they can induce, as here in this case, this is enhancer as well, looping of DNA. And this looping of DNA, as I described here before, it's important for controlling expression of the gene. So it means that without this looping, you don't have expression of some genes. If you express enhancer sequences, then you can loops, and then this is enough to stimulate transcription. And then it's the most strange thing, I think, but I have to tell you this. This is example, again, not important, the gene is not important, but what's important that next to this gene, in second orientation, in the different orientation, opposite orientation, is long non-coding RNA band. And actually, the problem with this is, to describe it, is because it's enough to start transcription. Long non-coding RNAs are not important at all. They will not bind anything. It's just some fact of transcription will open chromatin structure and expose enhancer elements which will activate the promoter of this band 4 genes. So it's not that it can catch some proteins, can bind to DNA. Sometimes it's enough to have transcription of it to change the structure of chromatin and then to activate promoter of neighbor genes. Okay, and this situation, maybe I will skip this because it's too difficult, and then again you can recognize that this is a plant examples, and this is so important, flowering, I mean long non-coding RNAs controlling flowering. So vernalization, do you know what it is? Yeah, so in some plants to flower, they must have lower temperature for a moment, for hours, days to flower. And this is the case that the flowering in plants is controlled by FLC protein. This is a negative inhibitor of flowering. This is negative inhibitor of flowering. And in warm weather, when the weather is warm all the time, the FLC RNA is produced all the time. Okay? You have two long non-coding RNAs encoded actually in the gene of this FLC. Cold air, a cooler. And actually cold air in this situation, conditions, is not active. But cooler is active, but it can form just the stem R loop. It's not clear here, but here should be RNA. And this R loop is recognized by NDX proteins. It doesn't matter, but nothing changed. The active FLC transcription is active. But if you have a cold for several days, then the cold air is long non-coding RNase is activated. And this long non-coding RNase is recognized by some protein which will methylate actually DNA at the FLC promoter and stop transcription of FLC. Again, the cool air in addition to this, so this is one regulation, but then the cool air is activated in the cold, and it's transcribed, and as an antisense, RNA is blocking actually transcription of it. So without any interaction, it's just blocking as an antisense RNA the expression of FLC. So you have two, in some cases even, I have some examples with 3 long non-coding RNAs within the same protein genes to control. Particularly in plants, it's popular, this kind of regulation. Okay, and the last part is the controlling post-transcriptional RNA processing. We will have a lot of examples, but just a few and very simple. So as I said, the long non-coding RNA can bind to some proteins involved in splicing. And this is, if it can bind proteins involved in splicing, splicing is removing of introns, then introns of certain genes will be stopped or regulated. Can be regulated, can be stability of RNA. For example, to RNA, some proteins can bind. In this case, is pumilo, the protein which will stimulate degradation of mRNA. But when NORAD, long non-coding RNase, appears, it will bind to pumilo and will sequester pumilo from the solution, from the cell, and then RNA will be more stable. It's very complicated, actually, situation when long non-coding RNA fast controls signaling pathways. It means that with this long non-coding RNase, beta-catenin is not degraded here in this complex. It moves to the nucleus and starts transcription of pluripotent cell genes. So it's not only that it will influence one gene, you know, it can influence a lot of genes by activating one transcription factor. And this is the last examples. Can interact with other RNA. You saw this example, the Stauffen-1 protein, and you have two different long non-coding RNAs, two different. And in one case, it will stabilize transcript, and in one case will actually move or push this system to degradation of the mRNA. Look at this. This is a really fantastic example. And finally, it can modulate mRNA translation. So in cytoplasm, long non-coding RNAs can interact with mRNA and can stimulate translation. And I said that this is the last one, but this will be the last one. In some cases, the long non-coding RNAs can bind to microRNA. Here is the example from mammals. I will not describe actually how these peanuts are produced. The peanuts, long non-coding RNAs, can form complementary sequences with miR20S, and the target, and you know that microRNA will recognize target mRNAs, and in this case will inhibit its translation. Without this microRNAs, you will have a stable target mRNA. Okay, that's it. I think that I will show this, just to show, to finish this. Long non-coding RNAs are extremely important for development and diseases, health, and whatever, of animals and plants. And you can see here, mouse, that if you deleted some long non-coding RNAs, something is going on. You can see difficulties in brain development, dosage compensation and growth, and you can see that in all cases that there is no process that are not involved in, that are not controlled by long non-coding RNAs. And you can see here by the red dots, I just put these names where we describe the mechanism directly during this lecture. And of course, long non-coding RNAs are very important in cancers. We know a lot of long non-coding RNAs characteristic to different cancers. So it's believed, and I think people now think that studying long non-coding RNAs in cancers can give us better characterization and diagnosis of particular cancer examples. And the same is true for plants. You can see I put here different processes regulated by long non-coding RNAs. And again, I put in red dots at the position of at the RNAs. We described mechanism and function of it. It was a short lecture, but I think we described a lot of different mechanisms. And just take home message, long non-coding RNA, biogenesis and functions. They are transcribed by polymerase II. Usually they contain the cap structure and poly-A tail. In most cases, they act in the nucleus, but in some cases, they are transported to the cytoplasm. They can be involved in, be like a sponge for microRNA, so you can form complementary sequence with microRNA. They can regulate modification of chromatin. We described these examples in several times. It can be kind of scaffold. It means that it can be, it can stimulate microRNA, long non-coding RNAs can stimulate interaction of proteins together. Then it can activate transcription or inhibit transcription in both cases, and then we describe this. And I think this is the end. Thank you very much for your attention, and if you have any question, I will answer them, if I can, of course. Thank you.